your part of the country, though I will be talking today is a workshop, so I am aware of that fact that it is a workshop, and so we'll be talking, taking a few steps back and try to build a few things. There will be not much details, and if anything, I'll probably avoid the details, but I'll give you the references. And my email address is quite available. You can get it in the internet, or otherwise there is one. Use my official address, which is bbagchi@iscac.in, and you can write to me for the details. So these I'll do several things today. Try to go through the basic idea is what can certain basic concepts in physics and chemistry combines with computers to play a very, very important role. Actually, many times it helps to take, as I said, take a step back and look at the very beginning. And so that's where I'll start. So this is a famous saying quoted by many people, highly cited article. I took it from, but this main thing is Van Dyer, right, 2007. You can get it. He said the next industrial revolution of designing drugs by computers, that became hugely important thing. In India, it is in Astra. In Institute of Science, a lot of that went on, but we also had next door Astra, which is now AstraZeneca. Astra had a huge group on the computer-aided drug design. And I knew some people who did PhD in exactly my subject, which is statistical mechanics, and they worked in that area. And this is CAD, and this computer-aided drug design is a very important area of research right now, and which is the reason why this workshop is so important. So there are many ways of looking into it. One of the way to look into it is that when I talked with my colleagues in Astra, that what they are doing, they are saying, okay, by computer, they are cutting down the possibilities that are there, because they are doing experiment, time consuming and very expensive. So, so one can filter through libraries and a smaller set of predicted active compounds, then that can be extended, and that tremendous save of money and human effort. So, but what lies at the basis of CAD? What lies at the whole design and help of computer? So these are the things that what we want to know. What are the theoretical and conceptual tools that forms the basis of computer modeling? How did it get started? Okay, then the basic things that we need are thermodynamics, like we need almost all the time doing free energy calculations. We need statistical mechanics. And the statistical mechanics actually gives you thermodynamics, but it goes a little deeper. It starts with the force field, how molecules interact. And there are two computer things that we use, which all of you know. One is the molecular dynamics, which started with Alder, Wainwright and Rahman. And then Monte Carlo Metropolis, which directly gives the free energy. But it is a bit easier said than done. Getting free energy and all these things are not easy in a complex system. And still, it's a very active area of research and lots of work going on. Many of the people, thousands of people in chemistry, physics, biology, working on this area, also material science. However, what today's lecture, I'll try to do use molecular dynamics of Monte Carlo, which I'll hardly talk about. That means I just say that this is obtained by simulations. My emphasis will be on bringing you the basic approach, telling you there are certain very far-reaching general uh, tools or far-reaching general ideas, far-reaching general insights that you need. Because remember, the more complex the problem is, more insight you need. And it is a really complex area. Okay. So again, it all started as a very interesting small story. Uh, in 1991, before most of you were born, I was visiting a great scientist, Robert Zwanzig, and in NIH, National Institutes of Health. And then I was in demand for a very strange thing by the people who are working on drug design. NIH, there's a huge amount of that research goes on. And I was in demand, within the first 2-3 weeks, a couple of groups contacted me and wanted to talk to me, came to meet with me, want to give me talk. What it turned out, they really wanted to use a thermodynamic perturbation theory, which was developed by Zwanzig in 1954, but everybody was very afraid of Zwanzig because he was a very temperamental person. So they could do the next best thing, which is they came to talk with me to see that whether how they can implement and where the difficulties they're having in implementing. And it's a very beautiful scheme where that allows you the free energy of a change. That means if I make a mutation, and then as a mutation, the free energy, will it increase or will it decrease? If free energy decreases on mutation, then that is good for you. You are looking for a binding, and that binding will then be favorable. So there are two things. One, you want to get right direction, and also that has to be also the active. That means it should be relevant. So thermodynamic perturbation theory is based on statistical mechanics, and that's a beautiful thing. That's in my book of statistical mechanics is done in a big way, and many other books has that. That's the one fundamental thing that we use in drug discovery. And then the structure property correlation is another important thing where X-ray comes, and then you get the structure. Then from the structure, you now introduce interactions and you try to get the property, like polymorphs, in which are so important in, say, for example, paracetamol and other things. All the drugs that we have, some polymorphs are active, some are not. Some polymorphs come and go. And then there is the enzyme kinetics, which is extremely important. Unfortunately, I'll not be able to talk on enzyme kinetics. So we have done a lot of work on enzyme kinetics, but many people are doing, even in my our extended group, people working on enzymes, understanding enzymes, protein dynamics. This area was given a Nobel in 2014 of the work that was published in 1976 by Warshel Levitt. Then also, I'll talk a little bit of drug DNA interaction. That there is chemotherapy, the basic chemotherapy. So, and then there are docking studies, a lot of going on, protein folding, allostery. The list is endless. Now, if you could look at it, what am I doing? What I am doing is the following. I am going to try to bring you not the very details, but want to give you what is the way to think about it, what is the way to go about it, and what are the things to look for. What are the things that are important in my opinion, of course, and they are very, very interesting. Then they're very interesting physics and chemistry in this, and mathematics in this area, which you better understand, because only by understanding you can get. Just like Zwanzig's 1954, and Zwanzig never thought that his theory would be used in drug design, and he had no patience for that also. So many of the things in fundamental science that come, if you know, you will be able to better appreciate the so always keep an eye on these basic fundamental things. There are a lot of beauty in that, a lot of beauty in this field, into the complexity, but that beauty can be appreciated only when you look at from the physics and chemistry, from some fundamental concepts. So talk outline, I'll start again, I'll go through something will be a little more deeper than other, and some I'll just a through. So these slides are largely made by my two students, who will be referred many times, Mr. Shomok Mukherjee and Mr. Shantan Mondol, and they submitted PhD thesis. So many of the things made metformin is done by that. Curcumin done what my student Milan Hasda. Vitamin D by another person, Susmitara, who is a faculty now in ISR Kolkata. And insulin, hexamar, Shomok, Shantan, also Pooja, who is in Chicago. This is the kind of work, and we have done many other, which I'm not going to talk on. But the idea will be fixed, the talk will be pedagogical, and so details will be omitted, as I am mentioning many times. Okay. So drug DNA I said it's a huge interact. Many cases. This is important, but one of the things is essentially cancer therapeutics, because this when it goes into you can intercalate many of these drugs, and then DNA stops functioning. The basic idea is that in cancer, within cancer cells, there's a lot more activity. So the DNA is opening up and closing much more, and there is a more chance of getting higher concentration of one kind of molecule which can go in DNA. So the search is finding that kind of a molecule which can go and put inside DNA. Now we start thinking, with you knowing anything, what kind of molecules I need, which will do this job. You immediately know one thing, that DNA has a negatively charged phosphate backbone. So I need a molecule which has a tail or a region which is positively charged, so that my positively charged things can go and anchor. And electrostatic interaction is a very strong interaction at a local level. Then you need it to go inside. Now we know there are these 4 bases in nucleotides in our GCAT, and so I want something which is flat, because those are flat, and we know how the DNA coils around in a helical form. So helical so it has to go inside. So you need then a guy which not only have a molecule, which not only have a positively charge, it will also need a flat regions. And what are the best candidates of flat region? Aromatic rings. So most of the cancer chemotherapy drugs consist of these kind. These are donomycin. Then actinomycin is there. All these guys are very similar thing. You look at the positive ammonia, protonated ammonia ion there, and you can see the flat things. So the way it goes on is like that. It just intercalates. Now the question there is what we asked and we studied, and it's when we started working on that, this is a beautiful problem, but there are few thermodynamic studies, but no detailed studies of what is the binding mode. How does a DNA goes, a drug, donomycin or actinomycin or cisplatin, these are very well known things we give to people all the time. So there are first generation, second generation, third generation things, but all has the same structure. So basically you want different kinds of drugs for different kind of DNA, and different DNAs are implicated in different kind of diseases. It's not a very pleasant subject, so I'll not go into that too much, but this is the basic game plan. So then what one does, one say, okay, I want to know where does it goes, and then comes a very interesting there was some conjecture on that how does it go? How does this donomycin goes into a DNA? So computational analysis, that means you do a simulation, and you essentially get the DNA, construct the DNA, and then called targeted MD. You pull the molecule, the donomycin inside, and then you do a what is the best way it is going to fit in, how it is going to approach. We find that it went and binds to the minor group, and much of the time in the 80 minor group. And then there is a really interesting thing is that when a drug will go in or not go in depend very much. Binding part can be controlled by size and many other things, but then there is a critical region, there is a critical region where it goes over, and that barrier, and then it intercalates. So this intercalated state is a deep free energy minimum, but there is one more slightly less stable free energy minimum, which is the minor group binding. Okay, so this is a very important piece of information if you want to design the next generation. You have to take this into account if you want to generate, and it can, this is the way you can start playing the game now in your drug design, that okay, this is what I have to do to design a new chemotherapy drug. Now I'll go over to talking this little on insulin, and there is a lot of very beautiful story in insulin. It is becoming a very big, actually, again, when we started around 2014-15, there are very few studies on insulin. And I remember I wanted to work insulin when I was in, first paper came out from our group by Kushal Bagchi and Sushmita Roy, and that is already reasonably well-known work where we just studied an insulin solvation. We are not interested, at that point we didn't know. But then I get to know that insulin has a lot of problem. Insulin aggregates. You cannot store insulin, and you have to store it very carefully. And there are a lot of, so I was visiting Rice University, and this guy, a protein folding guy, Professor Peter Wellness, I told Peter that it is, he had a computer program, can we study insulin aggregation or association, insulin association? I didn't know anything other than that. He said, Biman, that's a good problem because there's a money in that, because pharmaceutical industry will be important. However, we didn't get anything done there, but it stayed with me. Then these students, Shantan, Pooja, and Shomak, they did a wonderful job, and we collaborated with experimentalists to get some beautiful story of insulin, which is now quite famous and well known. And you'll see this the power of computer and power of reasoning and thinking in getting some beautiful thing done. Okay, so let's get started. So insulin is biologically active as a monomer, but it is stored in pancreas as a hexamer. Coming out of hexamer, it becomes a dimer, and then that also has a biological role, but then it goes to liver cell. There it becomes monomer. It is a monomer, and insulin is an extremely important thing in inflammation, many things, not just diabetes, in cancer. So there's a huge number of things which it stops, laid up things it promotes, and there's a list of things. I must have missed 100. I'm giving 10 or so. Okay, so now we'll get into the details of the molecule. And as I said, it's a biologically active, which is two chains, A and B, which are connected by disulfide bonds. So that gives a semi-rigid structure. However, then it, as I told you, that if in our body, insulin is stored as a hexamer. And coming out, it becomes a dimer. So we asked two questions. A, what stabilizes hexamer? B, how do we go from hexamer to dimer? C, how do I go to dimer to monomer? Okay, so we made progress in 3 of the 4 things that I'm going to talk. Now, when a dimer, so first I'll talk of the dimer, then first I'll talk of hexamer, then I'll come to dimer. So here on the left, I show the dimer. Hexamer is made of 3 dimer. That's why the left is shown. That there are chain B and chain A, and this is the monomer itself. This monomer is already very complex, 51 amino acids, and chain A and chain B stabilized by you can see the disulfide bonds are there, 3 disulfide bonds. They are given by elliptical circles, and in addition to disulfide bond, this is stabilized by hydrophobic interaction, and there are hydrogen bonds in the backbone. Okay, so now we'll first talk of the insulin hexamer. There's a first one started looking at hexamer. We find that the big cavity at the center. That was already a bit suspicious. Why is the cavity doing, and what is the role of the cavity? So 3 dimers form hexamer, and 3 dimers retain the dimer structure. Now this hexamer itself is a beautiful structure which has 2 zinc It can also be calcium. And then they are coordinated to histidine, and three histidine top, three histidine bottom, and there is a large number of water molecules. I just put N, and the question is that how many water molecules are there? That was the question. And this, you can see this beautiful structure as a storage unit, and that prevents segregation. If this structure were not stable, it could aggregate. So then we looked into the, we collaborated, as I told you, with Professor Gopal and his group in MBU, and then we got the X-ray done, and X-ray gave the kind of water molecules here. And these are a number of water in the simulation, so you don't have to pay any attention to that, but they shows 8 water molecules, and then our simulation also found these 8 water molecules. And this is the kind of structure. One cobalt is given 3 water molecules and 3 histidine. Very interesting structure. You know, from the point of coordination chemistry, is a very metallo-inorganic and metal-organic chemistry is very, very interesting. So first we looked into water dynamics. How many water are there? 8 or what? We found out no, there are several water molecules which intermittently going and coming, and they are little slow, not too slow inside, and these all bonds are breaking and forming. It's a very dynamic structure with a lot of entropy. So we wanted to now know how many water molecules. On the average, we found there are 10 water molecules, and these water molecules are different. They are exchanging, of course, but when they are in their place, we can study for a short time their rotation, and they are different. Like those which are bonded to zinc are slower, and the other 4 which are more rattling around, going A and A. And there it shows what happens if you remove the water molecules. So what is the role of stability of water? If you remove the water molecules, very quickly this cavity collapses, goes to a molten globule state, and useless, completely useless state. So the day it was published, immediately got telephone calls from California. The CNN news journalists got in touch with us, and they made quite a big story on that water holds insulin hexamer together. It was really a lot of fun. Okay. So the cavity summaries of this part is cavity waters are slow, long residence times. They weave intricate hydrogen bond network. And so we wanted to know one thing, you know, if you are taking insulin, for that matter, even if you are taking metformin, but insulin certainly, then you are not allowed to have alcohol. Ethanol is strictly forbidden. So we wanted to know that what happens if I have ethanol, and we did some calculations. So we found that ethanol gives rise to 2 different stabilized state, and like I shown there in the below, then ethanol molecules also coming and going and fluctuating, and we could identify this is the work of Shomok and Shantan, could identify 2 distinct states of ethanol, and they are different. So the state gets unstable. There is a lot of dynamics, but one binds little bit in the outside of the another goes more inside, but it distorts the structure. So once the ethanol goes in, and listen very carefully, once the ethanol goes in and install itself, it says a long residence time inside the cavity, much longer than water molecules. So your process of a hexamer going to dimer, will be in jeopardy. And this is an important lesson, that it screws up your insulin hexamer. So you might not get your regular supply of hexamer to dimer when the need arises. Okay. So they are again showing there are 2 structures that form. They are different from the ethanol induced structural, read the title, structural destabilization. More than structural destabilization, it get stuck in a metastable state. Okay, this has been also been published. So the summary is this: the final in ethanol, look at the final structure, it's completely messed up. And okay, now we'll do the other interesting thing, which is that monomer into dimer, again we'll study the effect of ethanol on ethanol, effect of ethanol on the insulin dimer thing. So here is the dimer shown the structure, the two monomers, yellow and green, and there are four hydrogen backbone hydrogen bonds as shown by dashed line, and then you have the hydrophobic contacts between phenylalanine, which are given on the right hand side, and tyrosine. So they are antiparallel. And now our question was that if when the dimer must dissociate when it goes to liver to monomer, how it becomes? What is this process? How the monomer dimer insulin dimer becomes monomer? We wanted to understand that. No study was there, not a single one. Actually, 6-7 also was not a single one. Now it's popular and it is becoming. These are kind of textbook kind of work. Okay, so we did free energy calculation, we did the role of water and other co-solvents like ethanol, and what with the conventional change of protein. So this is essentially what is doing that you have to pull it out. That means if two dimers are together parallel like this or anti-parallel like this, then I have to push them out, and that is very stable. So that's done by a technique which is called metadynamics, and well-tempered ensemble means you change increase decrease temperature and see that what are the you can pull them out. And this is a beautiful slide, I think either made by Pooja or by Sayantan, whatever. Now we are going to study the free energy surface of insulin dimer. Now there are two things that are important. One is the distance between the two monomers, center distance. I call that MM. How many contacts are there? In this case, there are a lot of contacts. Not only the hydrogen bonds, they form the native contacts, but also the contact between phenylalanines and tyrosine. So this is the native contact between them, which is in X-ray state is about 60. Putting into solution, bring it down to 55 or so. And so we did that. And then we, doing this free energy calculation, we obtained the free energy path. The free energy, what is the path it takes when it dissociates? This is a very interesting question. So number of contacts goes to 0 when monomers are completely dissociated, which is large RMM. So large RMM is the when it's going down. So number of contacts, as I said, is about 60. Then it comes down in solution itself. So this is the what you call A state, the deep blue state with about 50 contacts is the stable state in water at about 2 nanometer apart. Now, if you dissociate, it has to follow the following path because this is the minimum free energy path. So it goes from the deep minimum to large M even at 5 nanometer, 50 angstrom apart. Monomers. This is center to center monomer A. That means surface to surface will be about 30 to 30 angstrom. Even that far, they know each other. This is a very interesting thing. This is something completely new because the kind of theories people used to do before were turned out not correct. We are trying to do several elaborate or more detailed theories of this. So this is the pathway shown. That interesting is that now we're coming to association. You can look at association. They come when they come together, energy increases. And for a long time, no contact is formed, but molecules are coming together, but they are experiencing effect of free energy of each other. And I'll show you how. And then the path of ascent, if it is going to form a dimer, then descent, just like in chemical kinetics. And these are the many states that one forms. A state is the dimer stable state. Then it goes to stable B. It can get trapped in state C, which you don't want, but say B to D to E, F. That's the okay. And this is a free energy surface. One can calculate how does it the deep free energy minimum. Then it is a uphill, and then it recent temperature jump. This is the kind of time scales we get by doing some kind of transition state calculation for simple transition given here is barrier crossing dynamics that you know in undergraduate chemistry, physical chemistry. That gives a kind of time scale which are in pretty good agreement, but that is kind of fortuitous. I won't believe this number within a factor of 2. But it's still is a microsecond that the protein dissociation association taking place. They're very, very nice to know. So what happened to this when is ethanol? Again, we ask the question. And then when you have the ethanol, then ethanol becomes a risk factor for heavy consumption also in insulin. Okay. Mellitus, that's the wrong thing. That is a slide should come. So we did that. And again, one finds that the ethanol lodges itself in between, as it's shown here on the right side, that ethanol comes and gets in between because it has a big nice hydrophobic group in ethyl group, C2H5, and that really screws up the dissociation of, and it's shown the steps. It shows the dissociation of. It becomes more difficult to dissociate if you have fast. You can see dimer itself on the left hand side. The dimer itself becomes less stable now. Number of contacts is less. But then when you start dissociating in an intermediate step that's shown here, intermediate step gets more stable. So the deep minima that you have in the absence of ethanol disappears. Instead, intermediate comes in, and then it kind of hangs on there, and is difficult to get. So these are the kind of structure you get when ethanol goes in between two ethanol. So I can quickly go through. This is the big work which was published earlier this year, and references there. I'm not going to talk too much of that, but I'll just tell you some beautiful story in the water, because I just have 15 minutes more, so there are several other things to tell. Okay. So when I was telling you, even if there are two monomers, center monomers 50 angstrom or apart, surface to surface more than 30 angstrom, 30 to 33 angstrom apart, even then two in monomers. And this is a general problem of protein association. And protein association, protein aggregation is a big problem in neurological diseases. So when people start protein folding, they start talking as that if they get misfolded, then they aggregate. In order to, it have been designed such that they are stable in a monomeric form when they are in the native state, and you don't want them to the native state. In dissociation, if they're native state, they are difficult to associate. But those which form dimer, monomer, finds a way, dimer, tetramer, hemoglobin, they find a way to associate. And so the idea is that how does, and so there are always people have then one thing, that whenever proteins concentration goes beyond certain point, like all of us know it's more than millimolar, then it starts getting associated. So there is some cutoff. Why? That was the question. And so the idea is that, and also when they associate, they seem to associate, at least in case of insulin and many other, they associate in oligomeric proteins. They associate in a particular orientation, as if they know. What dictates it? What dictates the orientation? Who is controlling all these things? That is still an unsolved problem. So we looked into it, and we found that water plays an extremely important. So long range interaction between two monomers, even 55 angstrom, 5.5 nanometers separation, they are interacting. And so what we found is beautiful, my favorite picture, which is that there are this huge number of water molecules, and the water molecules inside. So when they are apart 5 nanometer, they are like that, they are coming close together. Look at 2 nanometer, when they almost from the native state, the water molecules just moving out. So in 3 nanometer is a critical region where there is these water molecules here actually surprisingly turned out to be faster than bulk. They are more mobile, but they control the association. So it is water control. So the it was that here is showing the insulin dimer, how it is fluctuating. It is hugely fluctuating, and that's the dynamics is really very, very interesting. So in ethanol again screws up the pathway, and so again the dimer dissociation is a problem. I am going to go to 5 minutes each in 2 other problems. One is metformin, and this is the standard, over-the-counter drugs, and this is the paper that came out, and is already fairly well-known paper. I see it's getting cited pretty well. Now, why did we started working on metformin? I'll tell you the story why we started metformin. Now, and also tell you a little story before that. It is the standard thing which marketed as a glucophage. Many people take it every day, and this is also again type 2 diabetes. So I was once having a breakfast, and used to have several times. I was visiting Senegal on some kind of a award and all.
Sir, your microphone got mute. You have to unmute, sir.
Okay, thank you. Okay, where did it get mute?
Just few seconds back.
Okay, so I can still. I was talking of the story of metformin. This person, I forgot the name, and thank you, Rajiv. And that person told me that old days it was so easy to find a drug because all you have to do go to a tropical country and dust a leaf. You will get a medicine or take a one of those these Ayurvedic medicine, and you can find the inherent molecule. But not anymore. Now we have to, in new things, we have to really do it by doing either biotechnology or doing computer simulations. And that really stayed with me, that the natural origin and the synthetic origin, the contrast that he made. So the story of metformin is pretty interesting. I know many of my colleagues take metformin, and one day I just took a look at metformin, and I was intrigued by the beautiful structure of metformin. That this is just amazing, that it has there is a C double bond N, another N. There are 5 nitrogen atoms and 2 methyl groups at the end. So it is amazing, and it forms in two planar forms, and these we found out by doing calculations, and then they are experimentally verified it. And there is a tautomeric, there is a form, bent form or twisted form. So these I'm missing not having the really molecular form. I'm more not the space fixed thing. I am the, but this is C double bond N, C double bond N, N on the middle and N on the left and N on the right. 5 nitrogen atoms. Beautiful molecule. And it is planar also, partly and partly the same bent. So then first thing was that one we got with an experimental colleague of mine, Professor Anindra Bhattacharya, and we did the UV spectroscopy. The reason is that we wanted to simulate this, and we wanted to know the characteristics of this guy. Not a single study existed. Maybe there are in some patents, but not in non-published form. So again, we had to find out what is the force field, and then Shanton primarily and Shoma, they did the force field calculation, and for that you need the IR spectra, and then quantum chemistry. Then you have to do melting of that. With that force field, you do the melting and find out how good is the melting, and we found out melting works pretty well, as is shown in the Wendy simulation. So now we were after we made the force field, we were ready to go, and we studied. We found some beautiful results. One of them is that metformin also acts as a chemotherapy because it blocks certain receptors. I think receptors of breast cancer. We wanted to its interaction with DNA, and yes, it forms a beautiful complex with DNA, and it binds to mainly to DNA minor group that is showing 80 rich minor group. It is such a simple molecule, has but beautiful, pretty strong binding. And this reference is given below. So, and he's shown the structure. This is again Shomok Shantan's work, and that it is binding, and then it flips from one to the other. We went on doing a lot of work on DNA. There is two beautiful work on DNA solvation dynamics that come out, long work, but don't have time to talk about it. And then interaction of DNA, experimental observation. We found that some CT spectrum, and there is absorption changes, and there could be some signatures of DNA-induced chirality, which we are still studying, and it's a very difficult problem to understand. Okay, so some more things. Now I'll be doing quickly one more thing. One is curcumin. This again, very interesting. I was in why Haldi is so important. Now, again, let me tell you 1 minute story. So this is the new thing. Internet is buzzing with this. When Jesus was born, then three wise men from east went with gifts. There are three gifts. Two other gifts were herbal things, and they are essentially some kind of a Ayurvedic medicine kind of stuff, or used also in Persia those days. But one, they gave gold. So people for long wondered why they gave gold to a new reborn person and to Mary, Mother Mary. So now the theory almost verified is that gold, they said, is nothing but gift of the Magi is the Haldi, is the turmeric that was given, because that is, we know, is amazingly useful as a medicinal properties. So we now want to know, and this now I am happy that I have the real molecular formula on the right hand side, and look at the beauty again. Two phenyl rings on the two sides, and there is this conjugated, extended conjugation. So now I want to know why. When in young age, I remember I used to have all kinds of problems in falling from trees, and all kinds of injury, and most of the time swollen arm and leg. Then my elder sisters of the locality, and they used to shout at me a lot, but then they put chuna no holud. My mother was going to college, university, and my father was so that was treated by locality those days. But chuna was always ready at home, and they used to quickly mix china and holud and put it. I still remember on my hand this holud. Why? I'm not go into the why mixture of these two, lime and haldi and turmeric. But I'll just talk of turmeric. Why it is anti-inflammation, and that's a beautiful thing, that it is antioxidant, all these things. The reason is what we came up, which is published now, which is again getting well cited. I noticed is the really of the book, the medicinal reason. They form an aggregate, very strong hydrophobic aggregate, very strong. And what we believe that the haldi, turmeric, forms these aggregates and excludes water. And if you want to get out inflammation outside your skin, that's why haldi is, we know, proven quality. Then if you can remove the water from that region, then your inflammation will go. Inflammation, which is nothing but bacterial infection, much of the time will go. So we studied that and see how they how the water dynamics here, how they form self-assembly, how the strength of the self-assembly, and that means this is we did these are big molecule, but we did up to 16, and they all assemble, and they assemble two ways, and they are parallel and anti-parallel. They are two, but look at the symmetry, and this aggregation, one can study the cluster size distribution, how well they aggregate. So usually all of them aggregate in a long run together, and you found what is a nano aggregation. And there are a lot of beautiful structures. Is what is called self-assembly. So hydrophobic, hydration-driven self-assembly of the turmeric. Again, turmeric also has a lot of anti-cancer properties, and people have used it to study, again, receptor blockage And you can understand that it's such a hydrophobic moiety can go and can block a hydrophobic part of the. So, but then we did the again free energy calculation, thermodynamics, the free energy of aggregation, then how in a 18 taking 18 curcumin molecules, what is the equilibrium state in a limited computer simulation run, and what is the largest cluster size. This I think is 16 mark. So the largest cluster size is here, which is not the most stable. Most stable is that when there is an octamer and these things, entropy, enthalpy, that is kind of a not a. So last little bit of thing, it is just 1 or 2 minutes. We did a lot of work on vitamin D. We published 2 papers, one plus one, another just came out in JCP. And this is something kind of important in the present day COVID-19 scenario. That what is vitamin D? First, vitamin D is your adaptive immunity. It is not innate immunity, it is adaptive immunity. That's why the same level as vaccine does to you. So this is known for a long time that look at 1849 that people knew that cod liver oil and all these things can help inflammation. And there's a Nobel Prize in this in 1903. And then unit sun for vitamin D. And this is the beautiful molecule again. And that's whenever vitamin D, I started, I looked into the molecule, and I'm really intrigued by this beautiful drug. Many things I want to do, but not been able to do. That what is the chemistry doing here? And since we have, before you couldn't do it, but now we have chemistry, we have quantum chemistry to give us intermolecular interaction, and we have molecular dynamics and Monte Carlo simulations. We are beginning to know these things, how the vitamin D will do. And this is the detailed kind of a semi-detailed biology that how vitamin D, the vitamin D3 goes to vitamin D receptor VDR, then how they interact with RNA to express many genes. Okay. This is a structure that is RX, another guy. Okay. Now, so what is the basic model? Basic model is very interesting. Here it is what we formulated. So if in order to kill the pathogen, I need a T cell, and you see a lot of this vaccine discussion, the T cell comes. Effector T cell kills the pathogen. But how do you get the effector T cell? You get the effector T cell from what is called antigen presenting combination of native T cell or sleeping T cell and antigen presenting cell. So these are the terms you hear these days all the time, antigen and all these things. However, it is like your native T cell, almost like a stem cell. It's a sleeping thing. Antigen presenting cells wakes it up and makes it a effector T cell. Effector T cell goes and kills pathogen. However, if you have too much effector T cells, then you are in trouble. Then you have autoimmune disease. You have lots of autoimmune, rheumatic arthritis, then psoriasis. Many things are, actually the difference between autoimmune is almost like a clock, but I'll not go into that. So in order to control then your affected T cell, you need what is called regulatory T cell. And that's where vitamin D comes in. So if you have low vitamin D, see, then you will have the autoimmune disease. And when affected T cell, the killer T cell, what is called killer T cell, becomes much more. You don't have vitamin D, so there is nobody to control it. So you get the cell. And so these are solved. This again, computer comes in, these huge equations. You solve. There are 2 things. One was the main paper is the +1. I don't know why the citation is not here, but the latest one came out earlier this year. So then there are a lot of interesting things. So you need basically healthy vitamin D, which is I think 50 nanogram per nanoliter or something like that. And to maintain a bistability. So on the last bottom picture on the right is important, that optimal vitamin D, you have a bistable region, neither weak regulation is autoimmune disease, strong regulation on the other hand will give rise to inflammation and cancer, many other problems. Okay. So this is actually weak is in one minima bistability is there for any stable thing you need, because it is performing two functions. It has to be bistable, and then strong it goes to T effector T cell concentration, and there is oscillations between them. We worked it out with how the structures with vitamin D, what without vitamin D. These are all published. I don't need to go into this, but we did certain amount of experimental verification of these things by whatever available. And as I said, this is quite an interesting. Yeah, original is given on the left side bottom, the plus one. So take home message is that role of vitamin D in preserving bistability is extremely important. So I am done. These work sketches, I put them on the top left. Shomok, Shayantan, and Pooja. They have done wonderful in these things. Sushmita, vitamin D. Milan started curcumin, looking very unhappy here. Collaborated in the metformin part with Ananda and student Rudra, and the insulin part with Professor Balasubramaniam and Ashish Desmukh, supported by JC Bose initially, now National Science Chair Professorship. Thank you for your attention. I usually have my books. Two popular level books I have written. These two slides were made by this put together by Shomok Shantan, except the initial part I wrote. So the two books that I wrote, one is the for students, which you can find by Googling, and it is available in Kindle, that how to